question. The part of the cell where respiratory proteins are found.
Inner mitochondrial.
Answer is inner mitochondrial membrane or inner membrane of the mitochondrion. Number 2. Which molecule evolved first in the timeline of biological evolution? RNA or DNA?
RNA.
The answer is RNA. Which characteristic of the nucleotide molecule underscore the genetic inheritance of either DNA or RNA? Nitrogenous base, hydrogen bonds, phosphodiester bonds, complementary pairing of nucleotides. The answer is complementary pairing of nucleotides.
I think Doc's gone.
No.
Wala yung name sa participants.
Yes. Gawa siya.
Hello, guys. I think something is wrong with the doctor's.
He might come back later. Let's just wait lang. Let's just wait for a little bit. Might be trouble with his connection in his end.
I'm out. Hello? Can you hear me?
Yes, Doc.
I'm sorry.
Yes, Doc.
I'm sorry, my internet at home is very unstable. I do not know what happened. Supposed to be that my internet is very fast here at home. I did not go to school today because I do not feel good, so I just stayed at home. So, can you hear me now?
Yes, Doc.
Yes.
Okay. What was my last question? Which molecules below may not be allowed to pass through the membranes? Water is allowed, oxygen is allowed.
Progesterone.
Glucose.
Glucose is not allowed. Glycerol is allowed. Carbon dioxide is allowed. Progesterone is allowed. So only glucose cannot be allowed in the membrane. Where in the DNA could be the site of recognition for transcription. UUU sequence, data domain, operator site, inducer site. The answer is
Tata.
the answer is data domain. What enzyme is responsible for the removal of introns in the hnRNA? Polymerase, nuclease, small nuclear ribonucleoproteins, nucleases, RNAs, DNAs.
Polymerase.
The answer is small nuclear ribonucleoproteins. What is the covalent chemical bond that we can find in the nucleic acids? Ester band, glycosidic band, amide band, phosphodiester bands, or nucleotide bands?
Phosphodiester band.
The correct answer is phosphodiester bands. What enzyme is usually used to conduct PCR in duplicating DNA? DNAs, RNAs, Taq polymerase, DNA polymerase.
DNA polymerase.
The answer is Taq polymerase. That's for PCR. What cellular structure is associated with modification of proteins? Golgi apparatus, nucleolus, nucleus, endoplasmic reticulum. The answer is endoplasmic reticulum. Kinsay naka-perfect? Naay naka-perfect? Na-perfect ba ninyo akong question? Wala?
Wala, Doc.
Wala, Dok.
Walay naka-perfect?
Wala, Doc.
Wala, Dok. Oy.
Sige. Walay naka-perfect? Okay, let's proceed to our lecture today. Our lecture today is the sequel of what we have. Can you see my slide now?
Yes, Doc.
Okay.
Yes, Doc.
What did we say about Milo interior? Is it the ICF or the ECF?
ECF.
ECF.
Yes, Doc.
So,
Easy, Doc.
the Milo interior is introduced by Dr. Claude Bernard, a French physiologist. In 1813, he died in 1878. He introduced the idea of Milo interior, which is actually the internal environment at which the cell is suspended into in our body. So all the cells in our body are suspended in the Milo interior, which is actually the extracellular fluid. And then what did we say about the homeostasis in our previous lecture? Homeostasis means maintaining the dynamic equilibrium of the cell. What do you mean by dynamic equilibrium in this particular case? It means maintaining the concentration of ions both inside and outside the cell. Take note that there are differences in the ionic content or the solute content inside the cell or the ICF and that outside the cell or the ACF. So when you talk about dynamic equilibrium, it means maintaining the concentration and the quality of ions suspended in the fluid of the interior and the fluid of the exterior. We know for a fact that the overall charge of the cell outside the cell is positive and the overall charge of the cell inside is negative. That is actually due to the compounding characteristics of all the ion contents inside the cell and outside the cell. The one that contributed so much on the negativity of the membrane potential inside the cell are the DNA, RNA, because they are basically negatively charged. And the third one are the proteinates or the proteins and amino acids. Those are the two molecules that contributes to the negativity of the cell inside. And I think the fourth reason why the cell is negative inside, it's because of the presence of what you call the potassium leak channel. This particular leak channel is a protein channel for potassium. Since there are more potassium inside, there is a tendency for the potassium to leak out from inside the cell going outside. Therefore, since potassium is positively charged, it will bring with it the positive ion. So therefore, when the positive ion leaves the cell, it will make the cell more negative because the positive ion is leaving in going outside. So in other words, the leaking of the potassium ions through the potassium leak channel is actually one of the contributory factors why the cell is negatively charged inside. So take note of those factors, ha? RNA, DNA, the proteinates or the proteins, and the fourth one is the leak channel. There is the difference between the potassium leak channel and the voltage-gated potassium channel. The voltage-gated potassium channel only opens when the sodium ion enters inside the cell. So that is, in other words, the voltage is changed from negative to positive, and that's the reason why the voltage-gated potassium channel will open and therefore will lead the way to rapid outflux or rapid outflow of potassium ion during or after depolarization or the influx of sodium ion. So take note that when the cell is stimulated, the sodium channel is open and it will now open the potassium voltage gated channel, which will now change the overall voltage inside the cell from negative, it becomes positive, and that is now the disturbance of what you call the internal milieu.
Doc is gone again.
Hello.
Hello, Doc.
Hello, good morning.
Hello, Doc.
I'm sorry, I was lost again. I'm sorry.
It's okay, Doc. Welcome back.
Okay, Doc.
Can you see me kay my slide or not
Not
yet?
yet, Doc.
Yes, Doc.
Not yet.
How about this one?
Yes,
Doc.
Doc.
Yes, Doc.
Yes, Doc.
Okay. So that's what I said. The most important thing that you should remember about physiological principle is that unless the cell is restored back to resting membrane potential, the cell is considered to be clinically dead. So when you say that the cell is clinically dead, it will not be able to respond to any stimulus, even drugs. Even drugs cannot get inside. So you cannot cure your patient if the cell of your patient is in imbalance. That's why the milieu interior has to be maintained at all times in order for it to be physiologically sound, or it is physiologically all right. So Dr. Bernard introduced the stable balance of the milieu interior, which is actually regulated by these different protein channels that are found in the membrane. So as in the case of the circulatory system, for example, the contents in the blood are distributed throughout the parts of the body, to the kidney, to the brain, to the lungs, and therefore these ions must be maintained at all times. When the excess water and excess salts are then removed by the kidney in order to attain that homeostatic balance. In the lungs, the carbon dioxide is discharged and oxygen is taken in, and in the digestive tract, all those nutrients are absorbed and they now contribute to the overall condition of the internal environment. So there are several feedback mechanisms in order to maintain the internal milieu. The first of all is the negative feedback. Accordingly, the negative feedback mechanism is a kind of response in order to promote stability. So for example, you are losing blood. So the negative feedback is that the blood, so when you are losing blood, normally the volume of the volume of blood in the body is, or the volume of water is also decreased, and therefore the kidney will now release renin in order to reabsorb water and restore the volume and eventually restore the normal blood pressure. So that is what you call negative feedback. In other words, the negative feedback mechanism promotes stability and restoration to that stable internal milieu, or the milieu interior. While the feed-forward mechanism is anticipating the change. So supposing there is a drop in blood pressure, anticipating the change is that when there is a drop in blood pressure, there will be drop also in blood circulation, and therefore low venous return, low cardiac output, and those are the corresponding responses for feed forward, anticipating particular change. While the positive feedback is actually promoting a change into one direction, instability, and it will lead to instability, and furthermore, it will lead to death or disease. One of the examples of the positive feedback is actually, for example, you are bleeding, and that bleeding results to decrease in blood pressure, and then decrease in blood pressure results to decrease in venous return. Decrease in venous return will decrease cardiac output, and decreased cardiac output results to decrease in heartbeat. And if that will continue bleeding, there will until the time when your heart will no longer pump. Therefore, that is what you call positive feedback. It leads to instability and therefore disorder or disease. So there is a difference between negative feedback and positive feedback. The negative feedback promotes going back to normal, while the positive feedback will now aggravate the situation and it will lead to one direction, and that is going to death. So for example, in the negative feedback, control of arterial pressure promote stability. So if the arterial pressure is dropped, then the sympathetic nervous system, the sympathetic activity of the brain will stimulate the heart to beat faster. So after that, there will be increase in heart rate and therefore vasoconstriction, and then lead to the restoration of the arterial blood pressure. So increase in heart rate will actually then cause the restoration, because that is actually the effect of the decrease in arterial pressure results to the increase in sympathetic activity of the brain, so that the sympathetic nervous system will stimulate the heart to beat faster in order to restore the blood pressure. That is actually a typical example of a negative feedback control of arterial pressure promoting back to stability. Another one is the baroreceptor complex, or the baroreceptor reflexes. The baroreceptors are very abundant in the aortic arc of our blood vessels as well as in the carotid arteries. So when there is a drop in blood pressure, the baroreceptors will send signal to the brain, to the sympathetic nervous system, in order to stimulate the sympathetic nervous system for the heart to beat faster, so that the blood pressure, or so that the blood volume will be restored to normal. So there is what you call vasomotor center that sets the normal points, and then when there is a drop in blood pressure, or the baroreceptors send signal to the sympathetic nervous system for the sympathetic nervous system to stimulate the heart, and therefore the heart will now beat faster, and therefore the blood pressure will be restored to normal, and it will be detected again by the baroreceptors, and it will send signal to the vasomotor to now stop the stimulation of the sympathetic nervous system. So that is what we call the baroreceptor complex, related to the negative feedback system that restores to stability. Another is the cardiopulmonary reflexes. So there is a cardiopulmonary receptor that will detect the cardio means heart, and then pulmonary means lungs. So the heart and lungs are actually coordinating to each other. So when the lungs receives less blood, so there is now less oxygen into the tissues. So therefore, the heart will now respond. The heart will now respond by beating faster and dilate the blood vessel and be able to restore the volume of blood and the amount of oxygen going into the lungs, so that after the lungs detect that there is enough oxygen, then the parasympathetic nervous system will work, so that it will now stop the sympathetic stimulation. The blood pressure is restored to normal, and the baroreceptors will now sense it, and therefore send signal to the vasomotor to stop the stimulation, so that the normal condition is restored to the optimal level. So that is what you call the feed forward control. So the feedback and the feed forward control, both the negative feedback and the feed forward control anticipates the changes, and the positive feedback promotes instability and eventually death. So in this particular example that you see in the slide, if you're a normal adult, you have about 5 liters of blood. So when you are under hemorrhagic shock, you are bleeding because of accident perhaps. If there is the loss of about 1 liter of blood, the loss of one liter of blood will send signal to the cardiopulmonary system in order to to regulate the blood pressure and volume of blood and then return to normal. However, if excess of 1 liter will be bleeding or you are bleed or having a hemorrhage exceeding 2 liters, after the 2 liters of blood had been lost, it will now lead to a positive feedback leading to death. So losing 2 liters of blood will eventually lead to the cardiac destruction. The heart will no longer pump and therefore it will lead to death. So that is the difference between negative and positive feedback. So here is a structure. So severe hemorrhage leads to decrease in venous return, and the decrease in venous return of the blood will also decrease the cardiac output. And decrease in cardiac output leads to the decrease in blood pressure, and decrease in blood pressure leads to decrease in coronary blood flow, and therefore it will lead to decrease in cardiac contractility until the heart will no longer contract and it leads to death. So that is what you call hemorrhagic feedback mechanism. So the action potential is actually another, this is a positive feedback. The cell depolarization leads to the rapid cell sodium permeability. Sodium gets inside, and when the sodium gets inside, the cell membrane potential is changed, and if that is not corrected, then the cell is clinically dead. If the sodium cannot be restored to the outside the cell, then the cell will be clinically dead, and it will lead to cell death. So this is another example of a positive feedback. So now let's go to the chapter 2, which is the cells and its functions. So let's now try to examine what are the salient features, the differences between the prokaryotes and the eukaryotes, because there are two groups of cells, prokaryotes and eukaryotes. So let's try to examine first the foundation of cellular theory. The cellular theory states that all cells forms the functional unit in all biological entities in this planet. So all living beings, all living things, be it bacteria, viruses, fungi, parasites, plants, and animals, consist of cells, which is actually the functional unit of every living being. And what is a cell? A cell, according to cell theory, the cell exists only in living things. So cells, only living beings have cells. Viruses are not living because they have no cells. So viruses are considered non-living because they don't have cells. So mRNA, like the COVID virus, is considered non-living outside the cell. They only become considered to be able to reproduce and multiply inside a living host. So that living being consisting of the cells, it is a fundamental characteristic of the cell that it can multiply by itself in the process of cell division. There are two types of cell divisions: mitotic cell division and meiotic cell division. Mitotic cell division is a kind of cell division that ensures the multiplication of cells. That, however, every time the cell divides, the cell will maintain its chromosome content in every cell. Take note that in a living organism, all cells contain the same genetic materials in all the cells. And those genetic materials contain what you call the genome of the total organism. So this is what you call the somatic cells. So the somatic cells are those cells that undergo the mitotic division. However, the second group of cell division is known as the meiotic cell division. The meiotic cell division is comprised by two phases. You call it the meiotic 1 and meiotic 2. The meiotic 1 is reduction of the number of chromosomes. So in the case of human being, we have 46 chromosomes. So after the first meiotic division, the resulting cells of the division of the first meiotic division can now reduce the number of chromosomes from 46 to 23 in the daughter cells, after the first meiotic cell division. The second meiotic division is known as equational division, meaning that those cells that have only half the number of chromosome will now be multiplied, but they will maintain the number of chromosome, which is actually 23. So the second meiotic division is known as equational, whereas the first meiotic division is reductional. So the meiotic division has two steps: meiosis 1, reductional; meiosis 2, which is equational. And the third corollary to the cell theory is that whatever happens to a single cell will happen to the whole body. So this particular third corollary introduced by Rodolphe Virchow is actually the basis why in medicine today, medicine and doctors endeavor to understand the disease at the cellular level. Because if you can explain the disease at the cellular level, you'll be able to trace up what is the cause of that particular pathological condition. So every pathological condition of the body can be traced into the single cell of that particular tissue in the body. Because according to the cell theory, whatever happens to a single cell, it will happen to the whole body. So that is the foundation of the physiological basis of medical practice. So here are the structural differences between the prokaryotes and the eukaryotes. One of the most fundamental salient difference between prokaryotes and the eukaryotes is the presence of nucleus. The nucleus in the eukaryotes, while the prokaryotes do not have nucleus. There are no internal membranes in the prokaryotes, or there are no internal membranes in bacteria. And the DNA in bacteria is single, while the DNA in eukaryotes is multiple and complex. The DNA in bacteria is circular, while the DNA in eukaryote is linear. There are no introns and no histones in the prokaryotes. So there are no introns and histones. By the way, introns are nonsense DNA sequences. So these are DNA molecules that do not code for any protein in the body. Remember that all proteins in the body are derived from DNA. And therefore, these are called DNA that are nonsense, meaning these are mere DNA sequences that do not code for any protein, or they are called introns. So you might ask as a question, Doctor, why is it that introns are there? My answer is this: as a Christian scientist, I believe that these introns was implanted by God into our DNA so that we will be protected from possible damage in sunlight. Because the sunlight has two ionizing radiations that can kick out DNA, or there are two radiation frequencies in the sun that is capable of destroying the DNA, and that is the X-rays and gamma rays. So this is the reason I would like to tell you today that do not overexpose yourself to sunlight no more greater than 30 minutes, because more than 30 minutes of sunlight exposure, you will be able to accumulate lots of these ionizing radiations that will tend to break down your DNA. So in short, what I have concluded in my study until this point in time is that I am very thankful to the Lord that the Lord has given us these introns in order for us to be protected from possible damage. Imagine that in eukaryotes, or in the human DNA, we have about 90% introns, meaning to say 90% of our DNA structures are not coding for any protein at all. So what is the significance of these introns? As I told you, if we are exposed to ionizing radiations, at least 90% of the time, the ones that being destroyed by sunlight are introns because they are 90% content in our DNA. So these 90% introns are some form of buffers or protective mechanism so that we will not mutate when we are exposed to sunlight. So when we are exposed to sunlight, 90% of the time, it is the intron that is being destroyed. So since introns are blank, they are not coding for anything. In other words, it will not affect us because there will be no changes in protein structure because introns are after all blank. They have no protein equivalent. So did you get that idea? Let us thank the Lord, our maker, our creator, for implanting introns into our DNA because that 90% introns is a form of protection from possible damage from ionizing radiation such as X-rays and gamma rays. Now, what is the significance of the presence of the histones? The histones adds up to the stability of the DNA in the eukaryotes because the histones is where the DNA or the genes are wind up. So yung mga the codes for protein are being held by histones in order to protect the genes from possible chemical damage from outside environment. So the histones will organize the genes and the DNA is wound up around these histones and they are now organized into domains. So those genes that are related to each other in one organ are packaged together in the molecule called histone. So for example, the proteins in your heart, the proteins in your stomach, the proteins in all parts of the body are packaged into one group so that they will be accessed immediately when it is needed. So it is the histone that actually package these genes together in one domain for certain parts of our body. Say, all the sets of proteins related to your heart will be packaged in subdomains so that it will be easily accessed by the enzymes whenever that particular protein is needed. I repeat, the histones organizes the genes into groups or domains that are organized according to the parts of our body. So in other words, the blueprint of our proteins are already packaged in domains, or they are clustered together so that when they are needed, it will be accessed by the enzyme in one setting. So the set of proteins that are packaged will now be unpacked by a certain enzyme so that when a particular cell needs it, it will be organized easily or it will be produced easily by accessing the histones. Did you get the point? So the histones are organizing molecules that will organize the genes into package of information inside the nucleus of our cells. So that is the significance of the presence of histones. Bacteria and viruses do not have histones so that they are scattered and they are easily destroyed by ionizing radiation. That's why bacteria, if you are going to expose the bacteria to radiation, they easily die. That's why after washing our clothes, we dry up it in the sunlight. Sunlight is an sterilizing agent because sunlight can destroy the bacteria and viruses in a very direct sunlight. So sunlight is a form of sterilizing agent because sunlight can kill the virus and bacteria because they don't have introns and they don't have histones. So they are easily destroyed by sunlight. So that is the significance of this. The absence of introns and histones in bacteria allow them to be easily destroyed by sunlight and chemical mutagens. So that is whereas our cells can resist this because our chromosomes are organized, they are packaged, they are protected, as well as they are provided with introns, which are protective mechanism in order to prevent mutation. Did you get the point? So in eukaryotes, the eukaryotes have true nucleus. It has internal membranes. What are the internal membranes that you can find inside the eukaryotes? One of these internal membrane structure is the nucleus. The second is the mitochondria. The third is the endoplasmic reticulum. The fourth is the Golgi apparatus. So these are the major internal membranes or compartments inside inside the eukaryotes. So the nucleus is enclosed in membrane. The endoplasmic reticulum is also enclosed in membrane, and there are two types of endoplasmic reticulum. You have what you call the rough endoplasmic reticulum, which is associated with ribosomes, and the other one without ribosome is known as the smooth endoplasmic reticulum. And as what I have said earlier in our pretest, that endoplasmic reticulum are organizing membranes that will allow the modification of proteins inside after the synthesis of protein in the ribosome. Because proteins are not functional until they are folded and they are modified in the endoplasmic reticulum. So it is very important that the internal membranes must be present. So you have the nucleus, the endoplasmic reticulum, both the rough and the smooth. You have the Golgi apparatus. You have the mitochondria is another internal membrane. You have also the microbodies, and you have the lysosomes and the peroxisomes. Those are structures in the inside the cell that have internal membranes. So another feature of the eukaryote is the presence of the linear chromosome. So our chromosome in the human body is linear, whereas in the bacteria or in the prokaryotes, it is circular. Our chromosome is multiple and they are complex. So we have about 46 chromosomes organized into 23 pairs of chromosomes. And we have introns. I have already elaborated earlier what introns are. Those are nonsense code that has no equivalent code for protein synthesis. Histones are organizing proteins. The non-histones are also proteins that will protect the chromosome from possible damage. DNA in the eukaryotes is complex and it has with telomeres. The telomeres are said to be DNA sequences which are synthesized by the enzyme called telomerase every time the cell divides. So when the DNA is duplicated by the DNA polymerase, at the end of the chromosome, the telomerase enzyme will now be organized or be activated and synthesize the telomere sequence. What are these telomeres? Telomeres are short tandem repeats of TG, or you call it the thymine and guanine tandem repeats, meaning to say sequences of about 6 nucleotides that are repeatedly connected to each other. You call it tandem repeats. So the same units like TTT, GGG could be a tandem repeat, and it is repeatedly several times in the telomeres, and each organism has a unique sequence of telomere. What is the role of this telomere? First of all, the role of the telomere is to prevent the tangling of the chromosome. Remember, we have 46 chromosomes. So in order to prevent the tangling of our chromosome, it is the telomere that will deter the tangling or fusions. The second role of the second primary role of telomere is that the telomere sequence will serve as the time clock for every cell tissue. So it was found out that every cell division, this telomere sequence is shortened. So after several series of cycles of cell division, the telomere sequence is shortened until such a time a critical number of telomeres have already reduced to a critical level. When the telomeres are already shortened to a critical level, it will no longer allow the cell division. So therefore, it will lead to what you call apoptosis or a programmed cell death. So every tissue in the body has their own time clock. For example, our skin has 28-day cycle. After 28 days, the telomeres are consumed and the cell will die. Our liver, for example, has about 200 days of cycle, and it reduces the telomere and undergo apoptosis. So there is a time schedule for every tissue in the body to die. our brain cells, it was found out that our brain cells will die every 600 days. Our liver will die every 300 days. Our stomach every 150 days to 200 days. So this telomere will be shortened every cell division. The shortening of the telomere leads to the death of the cell called apoptosis. So that is the significance of telomere. So there are two basic functions of telomeres. One is to prevent the tangling of the chromosome. The second is the time clock, meaning the reduction of this telomere sequence results to apoptosis. So other structures that are found in the cell of the living cells, there are structures that are called, in the membrane of a particular organism, there are proteins, there are phospholipids, and there are cholesterol that are arranged in a fluid mosaic pattern in all the cell membranes. So basically, all the membranes, whether it be prokaryotes or eukaryotes, the basic principle there is that there is a phospholipid bilayer in all the membranes. Whether it is a bacteria or plant or animal, the basic structure there is the phospholipid bilayer, which forms the fluid structure of the membrane. Gives this particular part of the cell, gives the cell integrity. So the cholesterol are found in between the phospholipid bilayer of all types of membranes. It regulates what comes in and what goes out of the cell. So the cell membrane is a very important part of the living cell because it regulates what goes in and what goes out from that particular cell. So for example, the kidney cell, the kidney cell will regulate what molecules to release or excrete out and what molecules to be retained into the blood in order to attain dynamic equilibrium in the internal milieu. So as what you can see there, the one that will actually regulate the flow of molecules into or out of the cell are those structures that you can see, the proteins, the phospholipid layer, the cholesterol, the sugar moieties. The sugar moieties are modifications of the protein to make specificity of every molecule that goes in and goes out of the cell. So the sugar is called glyco. That's why if that sugar molecule is attached to a protein, it's called glycoprotein. So if that sugar molecule is attached to a lipid, it becomes a glycolipid. And the role of this sugar moieties is for a specificity of the recognition of a particular molecule that would pass through the cell into or out of the cell. So that is the significance of the glycoproteins and the structure. So in summary, the plasma membrane have proteins. You call it the membrane proteins. You have the phospholipids, the cholesterol that gives rise to the cell integrity and the specificity of that membrane, so that specificity is very much important in the characteristic molecules that will go into or out of the cell. And take note, class, that each type of tissue in the body have different cell membrane components. So the membrane proteins found in the liver are different from the membrane proteins that are found in your skin or in your stomach or in your brain or all other parts of the body. So every tissue in the body has a unique characteristic of membrane proteins that will serve as what you call the histocompatibility proteins that are modified by sugar or called glyco. Glycoproteins are specified by the type of sugar attached to that particular protein. Or so in other words, the specificity of the protein relies on what modifications of that protein in their membrane are found. So that is the role of the glucose units. So you have here, there are two junctions in between the two cells that are together. There are what you call tight junctions. And in the tight junctions, nothing will pass through there in between. Whereas the second junction is known as the gap junctions. The gap junctions between two cells, it can allow the ions to pass through. So ionic diffusion between two cells passes through what you call the gap junctions. Whereas the other structure that are found in between the two adjacent cells is called the desmosome. The desmosome are protein fibers that will bind the cells together. So one cell bind together to a neighboring cell is connected to a desmosome or protein fibers binding it together along with tight junction and gap junctions. So take note that tight junctions and gap junctions and desmosomes are all proteins, and they are specific too. Now let's move on to the nucleus. The nucleus has a double membrane that separates from cytoplasm, and it has the nuclear pore. So the nucleus and outer membrane, and you have the inner membrane. The inner membrane of the nucleus is folded further into the endoplasmic reticulum. It has also the pores. Inside the nucleus are the DNA in the form of chromatin that clump up during cell division, especially during metaphase stage of the cell cycle of the cell division, the mitotic cell division. So the chromosomes are packaged into a tetrad during the metaphase. They are aligned up in the center, ready for the migration of the one set goes to the opposite side of the cell during cell division. The nucleus is considered to be the brain of the cell because it contains the DNA that controls all the structures of the proteins inside the cell. Remember that all proteins inside the cell are derived from the DNA that is contained in the nucleus. So the DNA contains all the genes responsible for the production of proteins, both the structural proteins and the enzymes that are present inside the cell are all derived from the blueprint, which is the DNA or the chromatins. So the nucleus is the brain of the cell, while the DNA is responsible for the blueprint of all, or the recipe of all the proteins that is contained in the cell. So although all cells contain the same genetic information, only those genes that are necessary for the production of protein unique for that particular cell type, it will be organized. In other words, the set of genes that is activated inside the liver are all the genes for the production of proteins for the liver cell. It will also be another different set of genes that are activated in the brain, another set of genes that are activated in the stomach cells, in the skin cells, in the bone marrow, and all the other parts of the body. So this particular set of genes, although present, all are present, only selected parts of those chromosomes are activated in a specific type of tissue. The gene is this amount of the DNA that controls the production of one protein. One protein, one gene, in a one gene, one protein theory. So every gene will be responsible for one protein. And these proteins are organized and are transported to the cytoplasm wherever it is needed. So as you can see in the right box, you can see the organization of the DNA. So basically the DNA is a sequence of nucleotides. And this sequence of nucleotides are organized in nucleosome. A nucleosome are organized into folded to a further domain, and furthermore until it becomes a tetrad. So the chromosome is a very intricate structure of the folding. So they said that our chromosome is folded about 10,000 times or more. So our DNA is a linear, but they are folded and are organized in the histones into domains and packets of information organized into a tetrad. So that is how our chromosomes are being packaged. There are 23 pairs of chromosomes, and each chromosome houses a particular gene, and the gene are packaged in one cluster, or they are clustered according to their structure in the body. So this is actually inside the nucleus. There is what's called nucleolus. The nucleolus is responsible for the production of ribosome. It is the dense chromatin in the nucleus. There is no membrane in the nucleus, but there is no membrane delineating the chromatin from the nucleolus. However, the nucleolus is composed of a dense pack of chromatin responsible for the production of ribosomes. So ribosomes are organized inside the nucleus, particularly organized by nucleolus. And the ribosomes are small clumps of RNA plus protein that is organized inside the nucleus, but they are dispatched into the cytoplasm. So after the ribosomes are organized in a clumps of RNA called rDNA plus proteins, they are now organized into ribosome, both big and small. The big subunit is measuring about 60S, and the small subunit is measuring about 40S. The S there stands for Swedberg unit, SVED, then Berg is Swedberg unit. So sometimes it's a sedimentation unit. So take note that there are two parts of the ribosome: the big structure, you call the big subunit, and the small subunit. The big subunit measures 60S, and the small subunit measures 40S. But accordingly, when big and small subunit will combine together during protein synthesis, the structure of the big and the small combined together becomes 80S. So this is the only wrong arithmetic in the whole planet, because ordinarily 60 plus 40 equals 100. But you will notice that when the big subunit and the small subunit will combine, it becomes only 80. So this is the only wrong arithmetic in the world. 60 plus 40 equals 80 when combined in eukaryotic ribosomes. I repeat, eukaryotic ribosome is unique. 60 plus 40 equals 80. So as I said, where does the other 20 go? It's called exergonic reaction. So it exists as a heat. So when the big subunit and the small subunit combines together, the heat is released. That's why the molecule is reduced from 100 to become 80. So 60 plus 40 equals 80 in the eukaryotic ribosome. Did you get that? Did you get that?
Yes, Doc.
Ang prokaryotic ribosome is measuring 50S plus 30S. So 50 plus 30 equals 80. But what happened is 50 plus 30 is only 60. So they are reduced. So that is the difference. So there is also another loss. So you can see there 60 and 40 subunit. The endoplasmic reticulum, as I said, there are two types. They have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. It's called rough because, as you can see in the microscope, the membrane is studded with the ribosome. So the ribosome is found associated with the membrane of the ER. You call this the RER, or the rough endoplasmic reticulum. Now, why is it that this rough endoplasmic reticulum is associated with the ribosome? Because it was found out that after the protein is synthesized in the ribosome, it will be packaged immediately inside the lumen of the endoplasmic reticulum in order to organize the protein to become functional, and of course the folding of the proteins or the folding of the peptides into a functional structure, and the addition of sugar, you call it glycosylation. So the process of adding sugar into the protein is called glycosylation, or sometimes it's called glycation. So glycosylation or glycation happens in the endoplasmic reticulum in order to modify the protein byproduct into a functional unit. So from RER, from the endoplasmic reticulum, this particular protein that has been modified is transferred to the Golgi apparatus for final packaging for it to become an individual vesicular products. In the contrast, the smooth endoplasmic reticulum, there are no ribosomes. However, it is the site of where the steroids are synthesized, the fatty acids are synthesized, and that's where you can find the fatty acids or the fats and oils, and of course the cholesterol. So the cholesterol therefore is synthesized inside the smooth endoplasmic reticulum. There are also enzymes that are present in the SER. These are what we call the toxins that inactivates the toxins and stores the calcium ion inside the cell, so that most of the calcium are sequestered in the smooth endoplasmic reticulum. And these packets of membranes that will transport the lipid from one compartment to the other as where the lipids are required. So the SER, or the smooth endoplasmic reticulum, is focused on the reproduction or the synthesis of fatty acids and steroids, as well as enzymes needed to inactivate the toxins. And these are the SER is responsible for storing calcium inside the cell. So you see the contrast. You see the in the left side you have the smooth, the rough endoplasmic reticulum. On the right side you have the smooth endoplasmic reticulum. So another compartment that you can find, membrane compartment, is the Golgi apparatus. You pronounce that Golgi, not Golgi, huh? Golgi apparatus is a packet of membranes that are called cisternae and vesicles, where the final packaging of the protein products happens. So after the modification of proteins in the ER, it is then pinched off and it is transferred to the Golgi apparatus for final packaging. They produce the membranes and lysosomes. They package the enzymes for lysosomes, you call lytic enzymes, and they also package the enzymes for the peroxisomes. So you have the microbodies and the peroxisomes. So you have this, you can see there the Golgi apparatus, and it is pinched off, and the packets of individual vesicles will now be called the secretory granules that contains the enzymes or protein products that when needed, it is discharged into outside the cell by the action of the calcium ion. So the release of these vesicle products into the outside the cell requires a calcium ion. You call it the vesicular traffic or vesicular transport. So every vesicular transport of this vesicular product requires high level of calcium, because it is the calcium through the calmodulin that these vesicles are mobilized into the plasma membrane, and it fuses there, and finally it discharges the product into the outside the cell. So vesicular traffic or vesicular transport in the process called exocytosis requires calcium ions. I repeat, calcium ions are needed for the transport of the products that are found inside the vesicle products after the products have been finally modified and packaged in the Golgi apparatus. So the lysosomes are examples of the vesicular products containing the hydrolytic enzymes that are able to digest proteins and bacteria and all others. Peroxisomes, on the other hand, is another vesicular product similar to lysosomes, but they contain what you call the peroxidases, or they break down the hydrogen peroxide. That's why they are called peroxidases or oxidative enzymes. So the lysosomes are found in the hydrolytic enzymes, while the peroxisomes contain oxidative enzymes. So you have the lysosome in contrast to peroxisome. Lysosome for hydrolytic enzyme, peroxisome for oxidative enzymes. The mitochondria is the powerhouse of the cell. It is also a double membrane, and it's folded. The inner membrane is folded to form cristae, where you can find the respiratory protein for the production of ATP. It contains also DNA and it is self-replicating. They found out that the origin of this mitochondria comes from bacteria. What is their evidence that it comes from bacteria? First and foremost, the DNA of the mitochondria is also circular, just like bacteria. The size of the DNA of the mitochondria is similar that of the size of the DNA in bacteria. And the manufacture of ATP in bacteria is also in the membrane, whereas the manufacture of ATP in mitochondria is also in the inner membrane of the mitochondria. So there are similarities between bacteria and mitochondria. That's why the endosymbiotic theory believes that in the past, the bacteria was accidentally enclosed by a cell, and that bacteria was no longer able to get out from that cell, and it becomes symbiotic of the cell, and the cell containing it becomes what you call the animal cell. That's why they have the mitochondria is derived from bacteria. The cilia and flagella are other cellular parts. These are projections from the cell membrane. And you can find that the cilia and flagella is composed of microtubules that are arranged in a 9+2 configuration. Now, why is it that the 9+2 was highlighted? Because the other cilia and flagella from lower forms, they are also microtubules, but they are not arranged in a 9+2. Why 9+2? You count the microtubules. You count the microtubules around the flagella. You have 1, 2, 3, 4, 5, 6, 7, 8, 9, plus 2 in the inside. That's why it's called 9+2 configuration. That makes a difference between the flagella of those of the paramecium or in lower forms. The flagella and the cilia of the lower organism is not in a 9+2 configuration. So that's why it is called, we have to differentiate the microtubules of the cilia and flagella in the eukaryotes and the cilia and flagella of the prokaryotes. Prokaryotes have no 9+2, while eukaryotes have a 9+2 microtubule arrangement. Did you get that?
Yes,
Another part of the cell is called the centriole. The centriole is a collection of microtubules in 9 triplets that are found in pairs. 1 pair equals 1 centrosome. And then the separate chromosomes, the cytoplasmic inclusion substances is stored. This centriole serves as the organizing center for the production of what you call the spindle fibers. So it is the cytoplasmic inclusion substances is stored in the cell-like hemoglobin, glycogen, and fat. These are extracellular materials that are found also in others. Centriole rather is responsible for the mobility of the chromosome through the structure called spindle fiber. The spindle fiber is made of microtubulin units, and these are the ones that are attached to the chromosome, and it allows the movement of chromosome during anaphase or metaphase, anaphase and telophase of the cell cycle or the cell division. So the significance of centriole is the production of the spindle fibers during cell division. And other cytoplasmic inclusions include the hemoglobin, the protein, glycogen, and fat. There are other also that are extracellular materials like collagen and elastin can also be found in the cells. Collagen are materials that serves as a matrix for the bone production, and elastin are proteins that lined up in the elastic fibers of our body, like the lungs, the trachea. All the other cells that have elastic properties have the protein called elastin. So those are the products. So if you try to look at the cell membrane is a fluid mosaic model by Singer and Nicholson. Singer and Nicholson proposed that the cell membrane is a complex structure of the cell wherein it has the phospholipid bilayer, which is the fluid part, and the proteins are floating. There are proteins cutting across from outside to inside. You call it the transmembrane protein. there are proteins that protrude in the surface. You call it the peripheral protein. So if the protein is attached to a sugar, it's called glycoprotein. If the protein is attached to the lipid, it is called glycolipids. So what is the significance of this protein again? As I told you earlier, the protein serves as channels, serves as receptors, it will serve as a carrier, it can serve as an enzyme or signal transducer in the signaling system in the body. Like the hormones are attached to the target through a particular receptor, and that receptor is a protein, a glycoprotein basically. So if you are going to summarize the function of the proteins in the membrane serves as carrier, it will serve as a channel, it will serve as a receptor, it will serve as an enzyme or signal transducer. It also serves as a histocompatibility markers. So let us summarize it. Histocompatibility markers, it serves as a receptor, it serves as an enzyme, it serves as a channel for transport, it serves as a carrier, and it serves for other purposes for the shape and structure of the cell are due to proteins that are found in the membrane. So this is the total summary of the structure of the eukaryotes. So the eukaryotes are basically different from the prokaryotes because of the presence of the nucleus, and the outer membrane of the nucleus is expanded into the endoplasmic reticulum, and there is the mitochondria, there is the Golgi apparatus, those are microsomal bodies, nuclear envelope, the nucleolus, the basal body, and the chromatins, the Golgi apparatus, and so on and so forth, which is not found in the prokaryotic cell. So this is the mitochondria. It is a double membrane also. The outer membrane is more permeable, while the inner membrane is less permeable. So the outer membrane is almost permeable to all the substances in the cytoplasm, whereas the inner membrane is a little bit rigid and impermeable. Remember that the mitochondria is the powerhouse of the cell, where you can find in the inner membrane the respiratory protein, which are responsible for the ATP production. We will discuss that in appropriate time later on in the oxidative phosphorylation in detail. I'm going to discuss that. Suffice it to say that right now, the inner membrane of the mitochondrion is responsible for the production of ATP for the cell. Remember this, 90% of the total energy that is produced inside the cell is manufactured in the inner membrane of the mitochondrion, which is now the powerhouse of the cell. So you have the nucleus, you have the outer envelope and the inner envelope or the outer membrane, and then inside it are the chromatin materials. So this is the structure of the chromosome organized into packets of information clustered using the histones and so on. So our next discussion will be on the transport of molecules into or out of the cell. Since we already have time already, so our next discussion next week will be centered on the transport
It's
of
9 hours.
It's the transport of molecules into or out of the cell next meeting, and then prepare for another set of quiz or a pre-test next meeting. Thank you very much, and thank you for indulgence. Did you understand now the differences between the prokaryotes and the eukaryotes?
Yes, Doc.
Yes, Doc.
Yes, Doc.
Yes, Doc.
Yes, Doc.
Highlighted in this discussion is the basic functional difference between the the DNA of the prokaryotes, which is circular, and the DNA of the eukaryotes, which is linear. Another important highlight is the presence of 90% of our DNA structure is intron or nonsense code, whereas in bacteria and viruses, they don't have introns, they don't have histones, and therefore they are easily mutated using the sunlight and spectrum of energy. Thank you, and God bless you.
Thank you, Doc.
Thank you,
Thank you,
Doc.
Doc. Thank you,
Good
Doc.
Doc.
Thank you, Doc.
Nice one, Doc.
I advise you to study very well because 40% of your lecture grade is derived from your quizzes. And our pre-test and post-test is classified under the quizzes. And there is no special exam in our quizzes. So yun ang importante doon. Huwag kayong mag-absent kasi walang special exam sa quizzes. Other examination have special examinations, but quizzes, we don't have special consideration. Pero mabait ako, kung minsan binibigyan ko kayo, pag maliit lang yung score ninyo, binibigyan ko kayo ng special quiz para dumami lang yung score ninyo. Ganyan ako kabait.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc.
Nice one.
Thank you, Doc.
So God bless you, ha.
Sir, I have one question.
What is your question?
My question is for our next pre-test, what are the chapters we will go through? I mean, the advanced reading.
Usually my pre-test
Chapter.
is either the chapter for that present lecture or the next succeeding 2 chapters ahead. So you better study the 2 chapters ahead because sometimes I get questions in my pre-test in those chapters ahead because I want the reason behind why I include in my pre-test the questions on the succeeding future chapters so that the student will read ahead or in advance. So
Okay, sir,
since
thank you so much. Thank you so much.
so you better study those chapters ahead so that you will be able for the pre-test. My purpose for that is that when you read the advanced chapters, it will now enable you to understand